The overall goal of this procedure is to use fluorescence to quantify the respiratory burst response. First, load the zebrafish embryos into the wells of a 96-well plate. Then, add a fluorescent substrate and activate the respiratory burst with a chemical agent. Then, quantify the relative fluorescence using a fluorometer. Ultimately, the fluorescence is used to show the respiratory burst potential.
This method can be used to study how experimental manipulations affect overall innate immune health. Here we use it to provide insight into the respiratory burst of zebrafish embryos, but it can also be applied to other systems, such as adult zebrafish kidney cells and cell preparations from other fish species.
Grow the embryos in deep petri dishes at 28 degrees Celsius in egg water until the desired developmental stage. Remove any dead embryos daily. Carefully decant the old egg water and replenish with new egg water daily. On the day of the experiment, make a reduced fluorescein working solution in a foil-wrapped 1.7 milliliter microcentrifuge tube. Also prepare a working solution of 20 micrograms per milliliter PMA final concentration. Then make 5 milliliters of each dosing solution in 15 milliliter conical tubes as described in the accompanying text. Keep the dosing solutions on ice.